Good morning. Very happy to see the number of people here at this early morning. And wow, the spotlight is on. Welcome to this morning's co-developed symposium entitled Looking Beyond VEGF Suppressions, Optimize Patients' Journey. So I'm Wai-Ching Lam. I'm from Vancouver. I have an amazing speaker experts this morning on the panel, and I will introduce them as they're going to present. But just briefly, we have Dr. Diana Do from Stanford, and Sunir Garg from Wales, and Dr. SriniVas Sadda from the UCLA and Doheny. So, don't forget our own national speaker, and Dr. Shaheer Aboobaker from Toronto. So Dr. Varun Chowdhury is the chair of the planning committee, and he led most of the kind of planning of these presentations. And unfortunately, because of the schedule conflict, he sent his regret he's not able to participate this morning's symposium. So here is my financial disclosure, and here are the financial disclosures of the other speakers at this symposium. And here's the disclosure of the commercial support. And more specifically, this program has received financial support from Roche in the form of an educational grant. And it's also received in-kind support from the Canadian Ophthalmological Society in the form of logistical support. And the speaker has received honoraria from the Canadian Ophthalmological Society. So, the learning objective of this symposium is that at the end of the sessions, we hope that you're able to learn the outline of the road of type 2 ENG pathway of the DME and neovascular AMD and how it may impact patient outcomes. They're able to identify the important biomarker on the structural OCT that can help patient how to predict treatment response in DME and neovascular AMD. And during the process, we'll also be discussing real-world case presentations and when to switch patient from one agent to another. Here is the agenda of the symposium this morning. And Dr. Garg will lead off with the presentations and discussing and showing us what's the emerging treatment of the neovascular AMD. And Dr. Sadda will highlight some of the biomarkers to help to predict treatment response to DME and neovascular AMD. And Dr. Do will update us on the faricimab in the diabetic macular edema and neovascular AMD. And myself and Dr. Aboobaker will share some real-life cases, and then we'll close off with the panel discussions, and it'd be great to have questions from the audience. So, I'm going to introduce our next speaker coming up. I was trying to decide how to go with their CV. And I got the advice from Dr. Sadda. He said, well, we can do it very quickly. You say, just say that we grab somebody from the street and put them on the podium and present it to the group. I thought that won't be an improvement. I actually know that's the response from Dr. Garg. But I would try to summarize it in maybe 2-3 sentences and with their long CV. So Dr. Sunir Garg is a professor of ophthalmology at the Retinal Service of Wills Eye Hospital and Thomas Jefferson University. He has published more than 400 peer-reviewed papers, and he is the deputy editor of the chief of American Academy of Ophthalmology One Network, and he is involved extensively in other like journal like Retinal Times, American Journal of Ophthalmology, Canadian Journal of Ophthalmology, and he's also the official clinical spokesman spokesperson for the American Academy of Ophthalmology. Just a little bit of side. He's been featured in Wall Street Journal, USA Today, and go on and on for the non-ophthalmological journals. So it is a pleasure for us to welcome Dr. Garg to share with us and give us the kind of what's coming up in the treatment of neovascular AMD.
Thank you very much, Wai-Ching. I appreciate the generous and kind introduction. It's a pleasure to be here this morning. My job is just to talk a little bit more about what's happening in this space before we get into specific compounds. So just a couple of questions for the audience. High dose of aflibercept, which we'll talk about in a few moments, can achieve 16-week dosing, and what percentage advised with neovascular AMD? 7%? Any 7%ers in the audience? Great, because that wouldn't be so impressive. 27%? That would be pretty good still. 47%? Any 47%? Half of the patients. Okay, let's go for broke. 77%. Anybody takers? We need more caffeine in this room. Oh, I got one. Very good. Number 2, tyrosine kinase inhibitors have the following attributes except good preliminary safety, FDA approval, US FDA approval for renal cell carcinoma, dosing every 2 years, sustained release of at least every 4 to 6 months. We'll talk about that a bit too. It's kind of amazing that 18 years after we first had anti-VEGF treatments available, our patients continue to do great. They do great with AMD, they do great with diabetes, they do great with vein occlusions, and the eyes tolerate them very, very well. This is a representative example of how revolutionary the patients are. Top and bottom is before and after. And the vision gets better, the anatomy gets better, we can also protect it over time. But what we've come to realize, though, is in clinical practice, we don't always see those kind of results, and it's hard for us to maintain those results. Part of the reason we don't see those results is because the patients we treat in clinical practice may not have been included in the clinical trial. Maybe they had subretinal fibrosis, maybe they had hemorrhage, maybe they had more chronic disease findings than you would otherwise expect. But part of it is it's hard for the patients to come in. Our patients are getting older. They have a lot of doctor appointments. We're not the only people in their lives, and fitting us in can be an issue. And then there's transportation issues with our patients. Often their adult child has to take a day off of work to come in. So maintaining the regular schedule can be hard. And even in the confines of a clinical study like the CAT trial, where at least in the US, there was no co-payments, you made it easy. Sometimes you got a lunch voucher. You could maintain those results for 2 years, but after that, the results started to peter out. And part of that is just due to the frequency of coming in. When you look at that data slightly differently, sort of the majority of that graph is the clinical trial data. And you'll see on average, it's a 1.5 to 2 line improvement in pretty much all of our clinical trials. But when you start looking at the real world data, we just don't see those results. It's about half of that for the reasons we just talked about. So because of that, there's been a lot of interest and a lot of focus in having more durable treatments. If I can have a durable treatment, I can bring the patient in less frequently perhaps, and then maybe I'll be able to maintain the visual acuity results better. There's a bunch of different things that are being looked at. One, of course, is faricimab, which is an anti-VEGF and Ang2 inhibitor. I'll defer that to Dr. Doe. I'll speak briefly about tarcocimab, which is the Kodiak Biosciences molecule. High dose of aflibercept, which will likely be FDA approved in the United States sometime in the next few weeks, and will likely be approved in Canada right around the first of the year. Touch a bit about tyrosine kinase inhibitors. And I'd also like to just introduce the concept of sustained release, including the port delivery system, biodegradable implants, and then touch a little bit about gene therapy. So Kodiak Biosciences has a really interesting molecule. It's an anti-VEGF plugged into this huge polymer. And the idea of this huge polymer is that it would just hang out in the eye for a longer period of time, and maybe that would give them a lot of durability. And some of the preclinical data suggested that it could be dosed as little as every 3 months or last as long as 5 months. So that's what they designed the trial to show. So if you look compared to aflibercept in the beginning, those first 2-3 injections got better, but then over time, the results started to peter out. They called this study the DAZZLE study. The results weren't so dazzling. And part of the reason for that is I think they were overly enthusiastic in what they thought the molecule could do. The other thing that you'll notice, it'll come up kind of periodically throughout the talk, is while a lot of patients do great without much, some percentage of patients, some around 10% of patients, really need treatments a lot more frequently, particularly for wet AMD. And if those patients don't have the opportunity to be given shots more frequently, those folks can kind of drag down the mean. And that may be what we're seeing here. There's a small amount of inflammation. We're obviously sensitized to seeing that nowadays since what we've experienced with other drugs, but the results were pretty favorable. There's ongoing studies for AMD looking at monthly treatment just to say that the molecule works. And then there's phase 3 studies going on for all the typical indications. And those will be coming out over the next number of months. The other thing that there's a lot of interest in is high dose aflibercept. So this is the exact same molecule that we've been using for a number of years, just more of it. So this study looked at aflibercept, which is our standard dose, every 8 weeks after a loading dose. Aflibercept, 8 milligrams, loading dose, then 12 weeks, and then 8 milligrams loading dose, then every 16 weeks. And if you look at the top line data, all 3 arms had very similar visual acuity outcomes, very similar change in baseline visual acuity. But what was exciting about this is patients were able to achieve those results with much less frequent treatment. At the end of the year, almost 80% of patients were able to be treated as infrequently as quarterly, with 3/4 being able to be treated as little as every 16 weeks. So 3 or 4 shots a year. So you can imagine why this would be such a big deal from a patient's perspective. They want to know, how long am I going to have to do this? How often am I going to have to come in? Well, so, it takes us a little while to get enough drug in the eye to get things going. But after that, maybe you'll be coming in 3 to 4 times a year. Once a season doesn't sound so bad for most of our patients. The safety profile was very similar to what we're used to seeing. The intraocular inflammation rates were very low in all 3 arms. And the stroke risk, heart attack risk, very similar with high dose compared to low dose, with no cases of retinal vasculitis seen in either dosage arm. So the other thing I like to talk about is sustained drug delivery. There's a lot of interest in this. The port delivery system was FDA approved in the United States almost 2 years ago now. Biodegradable implants, there's a bunch of different companies working in this space, and I'll talk a little bit about gene therapy. So the port delivery system was available by Roche and developed by Roche, and the study design was monthly ranibizumab, or the port delivery system, which is implanted surgically in the office, filled with ranibizumab at the time of surgery. It's a special formulation of it. And then the implant was refilled in the office every 6 months or so. And what you'll see is the port delivery system in monthly ranibizumab had virtually identical visual acuity outcomes, virtually identical OCT outcomes. So the implant worked really well. And if you ask the patients how they felt about this, because they were generally getting anti-VEGF injections prior to being enrolled in the study, they actually had an improved quality of life and actually preferred the implant compared to monthly shots. And then if you look at how many patients didn't require any supplemental injections, it was upwards of 95%. So it worked really well, and very few patients needed to get topped off with an additional in-office injection. There were some side effects that we saw with the treatment. Vitreous hemorrhage was an issue. The rates were quite high initially. We modified the surgical technique. Those results kept on coming down. And my expectation is that rate would remain very low as we gain surgical experience with it. Conjunctival erosion and retraction became issues, and that may have led to an increased endophthalmitis risk that we were seeing. But the reason it got voluntarily withdrawn from the US market is because of septum dislodgement. There's a septum or a valve, and when you refilled it in the office, you basically had to make sure you were going in straight perpendicular. If you went in at an angle, or if you started manipulating the needle, you could, in a sense, pop the septum or pop the cork, which would render the implant non-functional anymore. So the company's in the process of redesigning that. There's also a lot of interest looking at tyrosine kinase inhibitors. So this is currently FDA approved for renal cell carcinoma. There's a bunch of companies looking at different tyrosine kinase inhibitors. One of them is axitinib, which is the FDA approved treatment for renal cell carcinoma. And all of these are being delivered in some type of suspension or implant. I'll just show you one company. This is Ocular Therapeutics, but the idea holds true. So these are patients, who are both treatment naive, and all they received was a tyrosine kinase inhibitor. And you'll see anatomically there was a dramatic improvement initially, and that improvement in that first patient was sustained all the way out to a year or more. The other patient had improvement out to month 6 with some recurrence of activity. So biologically it seemed to work. This is all a phase 1 study. So these are patients who were frequent flyers, getting shots pretty regularly with our approved drugs. Then when they enrolled in the study, everybody got treatment with the Ocular Therapeutics drug. They got another anti-VEGF a month later. And you'll see about half the patients didn't require a supplemental anti-VEGF out to month 10. Some patients did, and some eyes just want to continue to receive anti-VEGF. That's compared to people who got aflibercept in the control arm. Finally, I'll touch base a little bit about gene therapy. This is sort of this hot topic. Patients always ask about this and stem cell therapy. Those are the two things they want to know about. And a bunch of different companies are taking an adenovirus, transfecting it into the eye with the hope of making ranibizumab or aflibercept from within the eye itself. Regenxbio Bio is farthest along. They have two different approaches. One is a subretinal approach. So we go in, do a vitrectomy with a microcatheter. We infuse the product underneath the retina. They also have another one that's looking at a suprachoroidal approach. So this is a result of the vitrectomy approach. This is a dose escalation study. Again, same idea. Beforehand, these were frequent flyers. We were getting a lot of shots. Everybody got therapy. And you'll see the yellow dots over timers. Who else needed therapy? Some eyes require anti-VEGF no matter what you do. But some eyes don't. And even as you go up and have higher dose, the number of patients requiring treatments overall goes down. The number of shots in total goes down. But some patients still require it. Some patients are going to have to come in for regular monitoring. The safety was as you would expect. There's some changes we saw were changes to the RPE, but we think that's related to the procedure itself, but maybe related to the adenovirus. This is the suprachoroidal approach. This is an in-office injection. This holds a lot of promise, and this is also being studied in a similar fashion. A dose escalation with this also shows similar results. Some patients didn't need any additional shots. Most patients required fewer shots. Finally, the Adverum has an intravitreal injection. The advantage of this is a very standard procedure that all of us have been doing for the past 15 years or so. This encodes aflibercept into the eye. Similar dose escalation study, a high dose up top, low dose at the bottom. The high dose worked great for AMD. It's really quite amazing how many patients didn't require another shot at all for 2 years. A few patients did, and that was really quite sporadic. The lower dose arm, some patients were injection free. Many patients had a reduced injection frequency. Some people still needed to come in. However, going forward, the arm that's going to be pursued is the low-dose arm. The reason for that is in the DME study, which was a virtually identical study, there was a high rate of inflammation and hypotony, and basically it's sort of a ptysis-like phenomenon in some of those eyes. So the high dose is no longer being pursued for either indication, and the company's directing their focus right now to the lower dose. So in summary, we have extended durability is kind of our new goal right now. We have longer acting anti-VEGF agents, including high-dose aflibercept, Kodak Biosciences, and then faricimab. We have sustained delivery devices. The port delivery system will hopefully be making a comeback someday. Tyrosine kinase inhibitors are going to be a hot topic we'll be hearing a lot about over the next couple of years. Gene therapy will also remain a hot topic. The other thing I think will segue really nicely with this is homo-CT. So as we were talking about gene therapy, some patients still needed to come in, but a priori, we don't know who those patients are. So I have to tell the patient, I can do this surgery and then you'll potentially not need so many shots, but I still have to bring you in for monthly monitoring. The value proposition starts to go down. I tell those patients I can potentially give you this therapy, monitor you at home. The machine will send up a flag if you have edema. We'll bring you in only if you really need to come in. All of a sudden, it kind of changes the paradigm. Thank you very much, and thank you to my colleagues for helping me with some of these slides.
Thank you. Thank you very much. So it's my pleasure to introduce my next speaker, Dr. SriniVas Sadda. I think I run into Dr. Sadda the first time in Halifax when we were traveling together at the symposium and we were on the ferry together. And Dr. Sadda is a director of artificial intelligence and imaging research at the Doheny Eye Institute. He's professor of ophthalmology at UCLA. He is the immediate past president of Doheny Eye Institute. And his research area include imaging analysis, advanced imaging technology. And he has more than 650 peer-reviewed publications. He's the editor-in-chief of the 7 editions of Ryan's Retina, like the Bible of the Retina. And he's also served as the editor-in-chief of Gravy's Archives for Clinical and Experimental Ophthalmology, and go on and on. But I think he's just the perfect person to give us this talks on biomarkers to help predict treatment response in DME and neovascular AMD.
Wai-Ching. Thanks for that very kind introduction, Wai-Ching. Okay, so I'm really pleased to be here. So I'm going to talk about the biomarkers that we use in DME and neovascular AMD, and let's get right into it. And this is sort of I'm going to start first with neovascular AMD, and this is really meant to be a partial list. I have limited time, so we'll talk about a few things here. Many of us who were treating patients in the PDT era, I'll call it, of neovascular AMD, we really had to get very good at fluorescein angiography classification of these lesions into occult, classic, and the like. And of course, now we've shifted to OCT in this anti-VEGF therapy era, but I'm going to argue that subtyping the lesions still matters, that occult CNV more or less is equivalent to type 1 neovascularization, which means below the retinal pigment epithelium, a classic to being type 2 between the RP and the photoreceptors, and type 3 being interretinal. And again, I'm going to argue that this is important because it has some prognostic importance. We recognize now that type 1 neovascularization may actually have a protective effect against the development of atrophy, and that actually the substrate for that appears to be the development of essentially a neochoric capillaris at the anterior surface of some of these membranes. It actually can kind of rescue the photoreceptors in RPE. At the same time, though, type 1 lesions are more prone to develop aneurysmal dilations or these polypoidal lesions. And of course, now we have consensus criteria for identifying these kinds of polypoidal lesions using OCT alone. Type 2 neovascularization, very important to detect because this can appear as subretinal hyperreflective material or SHREM on the OCT. And we have very good data that the persistence of this material, or especially if it's thicker, can be associated with worse outcomes. And it's interesting, and this is actually a long-term study we did of patients who were treated more than 8 years with anti-VEGF therapy, again showing that the patients who had the worst outcomes were patients who had thicker SHREM. So you might think that aggressive therapy to eliminate SHREM could be important. And in fact, we have some evidence to suggest that's the case. For example, in the Hawk and Harrier studies in the brolucizumab, it was the first time we saw one agent show superiority in terms of reduction of SHREM, greater reduction of SHREM in brolucizumab compared with aflibercept. And SHREM, again, was associated with worse vision in that study. Type 3 neovascularization, I find it very important to recognize these patients actually respond very well to anti-VEGF monotherapy in terms of reduction of exudation, but these patients invariably develop atrophy. And so that's why some people have even suggested maybe for those individuals that you might consider as needed therapy as opposed to treat and extend because of this propensity for atrophy. It's not only the type of neovascularization, but also the compartment of fluid that seems to matter. Of course, fluid can appear in the intraretinal, subretinal, or sub-RP compartments. Intraretinal fluid is generally associated with worse visual outcomes, no matter what study you look at. And some people have said, oh, that means you must need to more aggressively treat this. But on the other hand, it just might be a sign of a bad eye. It might be cystoid degenerative accumulation of fluid as well. But in any event, it is a bad prognostic sign. Subretinal fluid, of course, has been more controversial. Some studies saying it's bad, some it's good. It ultimately can't be good because we know exudation is bad. But one of the reasons that people thought that perhaps it was quote unquote protective, which I think is not a good term to use, is that maybe you might see less atrophy in this context. I think it's just a sign that you have a functional type 1 membrane. This is actually the study that we did from the Harvard trial that suggested that patients who had subretinal fluid actually developed less atrophy. Sub-RP fluid is probably the most difficult to resolve. It's most likely to persist. And sometimes it's the first thing that comes back when you're seeing a patient and you've quieted down their eye, you're looking for recurrent fluid, and sometimes can be a challenge, but some of our newer agents do a good job with this. This is just more data. This is actually data that was generated by Ursula Schmidt-Erfurth, and you're seeing two different curves in intraretinal, subretinal, or sub-RPE fluid. Regardless of the fluid compartment, patients who had more disease activity, more fluid, essentially, they ended up with worse vision outcomes. And just, I think, it highlights that regardless of where the fluid is, it is important, each of those compartments. Lastly, I want to note that it's not just the presence of fluid, but also the fluctuation of fluid that is important. This is a study where they divided patients into quartiles based on, there's some patients who basically you treat them, they get dry and they stay dry. Other patients, they can have these big oscillations. And it turns out these big oscillations are bad. You want to avoid that because those patients, they end up with worse vision outcomes. So I have a polling question based on what I just presented so far. And that's what type of neovascularization on OCT is invariably associated with the development of atrophy. So this is a test if you're paying attention early in the morning. So I guess you can go ahead and vote. Okay, I think we it looks like most people got it, so we can move on to the next. So now I'm going to talk about my second half about the diabetic macular edema, and again, whole bunch of stuff. I'm going to go through it kind of briefly and touch on these again. I don't mean this to be some people who feel that their favorite biomarker is not here. Please don't be offended. Limited time. So I'm going to hit sort of the highlights. And so there are also types. This is not type of neovascularization. These are sort of morphologies, types of diabetic macular edema. And so, there's been an evolution over time in the field, and kind of people have settled into these kind of broad categories now of patients who can have some diffuse retinal thickening. Probably these patients even have tiny cystoid spaces, but in any event, this is thought to be an earlier phase, and these patients generally respond well. Then once you develop cystoid macular edema, this is more chronic. Some of these may actually be degenerative, as I was saying with neovascular AMD, maybe due to Mueller cell injury. And finally, some patients can actually develop accumulation of subretinal fluid. We've all seen that in some of our diabetic patients, and some have speculated that this may be due to additional disruption of the blood-retinal barrier at the RP level as well. And maybe these patients may respond more poorly to anti-VEGF monotherapy. At the same time, though, even though subretinal fluid may be a negative prognostic sign in regards to treatment response, if you are able to reduce the fluid, several studies have shown that these patients can have significant vision improvement. At the same time, though, as I mentioned, there do seem to be poorer response to anti-VEGF monotherapy in some trials, and this is why there's been a suggestion that perhaps these are the kind of patients you might consider other therapies first line. Another entity that gets a lot of press, I guess probably less so in North America, but I tell you in Europe and Asia, there's a lot of fixation over this hyperreflective lesions in the retina, I'll call them. And there's a lot of confusion of the terminology because these terms are used inconsistently. But hyperreflective foci generally we think are larger type of lesions that have a similar reflectivity to the RPE. And you can see those big sort of spots there. They have posterior shadowing and the like. We think they correlate with lipid exudates. Whereas hyperreflective dots or spots, they're smaller. You can see the little orange arrows there, these little tiny lesions. And the reason it seems to be important to distinguish them, these smaller lesions may actually represent some type of inflammatory biomarkers. They may be activated microglia. And the thought is, in a variety of different studies, that there may be a poor response to anti-VEGF monotherapy in that situation. You may need to target other pathways. And in fact, other studies have shown that the more of these spots that you have, the better response these patients have to for example, dexamethasone. A couple of other biomarkers that I think many of you are quite knowledgeable about include drill or disorganization of the retinal inner layers. This is where you have loss of the clear laminations of the inner retina. Typically, it's because of ischemia that happens. And of course, a disruption of the outer retinal bands like the EZ, which corresponds to loss of photoreceptor integrity. So those are important biomarkers related to vision. Both are associated with worse vision at baseline. But one of the things that we've learned now is that they're not always irreversible. So you can actually have reduction in drill area and even areas of EZ disruption, and that can correlate with improvement in vision in patients. Vitreomacular interface disease, of course, is important to recognize in patients with diabetic macular edema. This itself can produce chronic traction, can produce an inflammatory component. But most critically, of course, we're identifying patients who may be surgical candidates if the edema persists after a trial of pharmacotherapy. And finally, most recently, people have really started to focus on the morphology and details of the actual cystoid spaces themselves. And some patients with cystoid spaces can have that kind of bright outer retinal wall. The yellow area is a little misplaced, I'm just realizing. And so that can actually be a sign of worse response to anti-VEGF therapy. So again, still early days for looking at some of these biomarkers. I did want to highlight, though, just very similar to neovascular AMD, it's not just the presence of these signs, but it's the fluctuation that matters. So patients who have more episodes of recurrent hyperreflective foci or subretinal fluid, two of the biomarkers that I highlighted might be negative biomarkers. These patients tend to have worse vision outcomes in the long term. So again, this is sort of immediate test of what I presented kind of thing. So OCT biomarkers associated with worse visual prognosis in eyes with diabetic macular edema include drill, EZ disruption, both drill and EZ disruption, or neither. So you can vote. Okay, I think we get the idea. Well done. Okay, good. People are awake, or the coffee's working or something. So, all right, so we'll move on to the close of my presentation. And so I just wanted to say, well, it's great that I covered a bunch of biomarkers. what does this actually mean for our patient management? Well, you might think that if you have these biomarkers that are associated with poor outcomes with anti-VEGF monotherapy, maybe you need to be more aggressive on these patients, right? Because we're all trying to figure out when do we switch, et cetera. These biomarkers, I use them in my practice on patients that I talk about patients as well, in terms of I might be worried about your long-term outcomes, things of that sort. But these are the kind of situations where you might consider earlier treatment. I put an asterisk on DME because I did want to emphasize the neovascular AMD biomarkers, I think, are been very consistent across studies, have a lot of confidence in them. With DME, there are some, I could probably for everyone show you a study that, may not be showing the same results. So that's why I put a little bit asterisk, but I think these are meaningful. So I was asked to talk a little bit about AI. It's something that I work heavily in, and where does it fit in? One thing that's good for us going forward, if we're starting to use these biomarkers, it's great in a busy clinic. If they can be quantified automatically for us, Sunir talked about home OCT, and those kinds of technologies will come with automated software to quantify fluid in these different compartments. So that's going to be nice. The same thing can be done for diabetic macular edema. There are pretty good segmentation algorithms out there to segment these individual things, as well as even things like hyperreflective foci, right? So if you're going to look at that for treatment response, I sure wouldn't want to sit in clinic and counting all of those hyperreflective foci. So to be able to do it automatically is going to be helpful. Also, you can train deep learning systems to try to make predictions for your patient. This was work that was done trying to use data from the Harbor trial and trying to predict how many injections the patient would need or their ultimate vision outcome. So that kind of stuff I think is going to be pretty useful for patients going forward. And you can do the same thing with diabetic macular edema. This was actually a agnostic study to try to figure out what features are important. And isn't it interesting that in terms of predicting outcomes in this deep learning approach, hyperreflective dots turned out to be the most important predictive feature. So I think that's actually, there's something there that's an important thing to look for in our patients. But ultimately, though, now most everyone here has heard of generative AI, where you can produce images and things of that sort. It's a little bit of small print here, but this is actually a generative AI model that you essentially predicted what the OCT would look like after treatment for this patient, right? So that's kind of cool. You produce this kind of a synthetic image. But where this is all going ultimately is, and again, I apologize for the small print, but this is a model system where you took your patient coming in, you put their images into the model, and it predicted this is what the patient's retina would look like on aflibercept. This is what this patient's retina would look like on ranibizumab. So this ultimately might be able to allow us to predict which treatment is the best for that patient. So this is kind of this concept of this era of personalized medicine. So to summarize, I've very briefly covered a number of different structural OCT biomarkers that can be helpful in predicting outcomes for patients with both neovascular AMD and DME. And these biomarkers, I think, are relevant to this discussion this morning because they may identify patients who are likely poor responders to anti-VEGF monotherapy. So when you're thinking about switching, I think these could be helpful signs to say, probably this is the kind of patient I want to switch. I do think AI approaches, though, are going to help us in terms of making these kinds of predictions as we enter this era of personalized medicine. So thanks very much.
Thank you, Dr. Sadda. That was a quick whirlwind tour of biomarkers. So I want to introduce our next speaker, Dr. Diana Do. She's the Vice Chair of Clinical Affairs and Professor of Ophthalmology at the Byers Eye Institute and Stanford University School of Medicine. And she has been a principal investigator and co-investigator for more than 35 clinical trials, investigating novel treatments of retinal diseases like neovascular AMD, diabetes, retinopathy, DME, vein occlusions, author of 125 publications. And it's perfect that she'll be giving us the update on the faricimab in diabetic macular edema and neovascular AMD. Thank you.
It's a pleasure to join all my Canadian friends this morning again and talk about an exciting new therapeutic option that was recently approved both in the United States and Canada for the treatment of neovascular age-related macular degeneration and diabetic macular edema. What are the key points of my discussion today? First, faricimab is the first bispecific that has been approved for use in retinal vascular diseases. Faricimab targets both VEGF-A and angiopoietin-2. Faricimab in phase 3 clinical trials was proven to be non-inferior to aflibercept in both neovascular AMD and diabetic macular edema, And faricimab was able to achieve these highly efficacious visual acuity results with fewer doses. In addition, faricimab has been shown to have an excellent safety profile. First, let's look at neovascular AMD. The Yosemite and Rhine clinical trials actually were in diabetic macular edema, and we'll look at that first. This investigated faricimab dosed every 8 weeks or used in a treat and extend model with up to 16-week dosing, compared to the gold standard, which was aflibercept 2 milligrams given every 8 weeks after 5 loading doses. So this figure illustrates the dosing regimen for this clinical trial. And we can see that both different regimens for faricimab allowed the clinician to either randomize the patient to standard every 8-week dosing of faricimab compared to a more personalized treatment interval, which might stimulate what we do in clinical practice, which is treat and extend. And the patient was able to move up or down in the personalized treatment interval based on OCT and visual acuity results. Let's look at these results. So when we look at both visual acuity and OCT anatomic changes, we can see that both faricimab treatment arms were non-inferior to aflibercept. And we see that there's rapid improvements in visual acuity during the loading phases, and this was sustained through year 2 of treatment. Let's take a closer look at the dosing regimen. We can see that 62% of faricimab-treated patients were able to be dosed 16 weeks during the study period. And in fact, 15% were able to be dosed on a every 12-week dosing arm. And the median number of injections through year 2 was for a faricimab personalized treatment arm in that second year only required about 3 injections. This is compared to aflibercept every 8 weeks, which required about 5 injections in the second year. What about fluid? Can we look at how fast patients had absence of fluid due to their diabetic macular edema? So this figure illustrates the absence of diabetic macular edema, and we can see there is a suggestion that if you use faricimab, patients were more likely to have resolution of their diabetic macular edema compared to aflibercept. What about absence of intraretinal fluid? Voss showed us that intraretinal fluid typically is associated with worse visual acuity outcomes, so it's advantageous to remove that. In addition, when we looked at faricimab compared to aflibercept, it suggested that faricimab was more likely to eliminate intraretinal fluid with fewer injections than aflibercept. And this was a post-hoc analysis. Now let's look at safety. That's very important with any new agent that's on the market. When we looked for rates of intraocular inflammation, there was no safety signal associated with faricimab. In addition, there were no serious adverse events such as retinal vasculitis or retinal artery occlusion. So it appeared from this safety data that faricimab was well tolerated. What happens over the whole 2-year period? Of course, we're always excited about what happens the first year, but when we look for durability, we want to look beyond the first year. And we can see that in diabetic macular edema, faricimab was shown to be highly efficacious with durability through 2 years of treatment and follow-up. Now let's move to neovascular AMD, which is another common condition we encounter in clinical practice every day. The Tenaya and Lucerne phase 3 clinical trials evaluated faricimab dosed up to every 16 weeks compared to aflibercept 2 milligrams dosed every 8 weeks. And we can see that in this clinical trial, the faricimab arm was able to be adjusted based on disease activity assessments that were criteria based on visual acuity gain or loss and also OCT changes. The primary endpoint of the study was a blend of weeks 40, 44, and 48, and this was a non-inferiority visual acuity trial. In the second year, starting at week 60, patients in the faricimab arm could go to a personalized treatment interval approach. similar to a treat and extend that we would do in our clinical practice. Now let's look at the results. First, at the primary endpoint at year 1, faricimab was non-inferior to aflibercept. In addition, when we go beyond the primary endpoint and we look at year 2, we can see that faricimab also was able to behave similarly to aflibercept, but with fewer injections. So let's look at some anatomic correlations and ask anatomic improvements in patients randomized to faricimab compared to aflibercept during the matched dosing phase, that is from baseline to week 12, because this is when both treatment arms received monthly injections of their particular agent. We can see that it's suggested in this post-hoc analysis that faricimab was more likely to result in the absence of subretinal fluid and intraretinal fluid during this matched phase compared to aflibercept. And what would that be due to? Is it the higher molar dose of ranibizumab that's in the faricimab, or is it due to angiopoietin-2 suppression, which is a new feature of faricimab? Among patients who had intraretinal fluid and subretinal fluid at baseline, more patients treated with faricimab achieved absence of both intraretinal fluid and subretinal fluid faster and with fewer injections than those patients treated with aflibercept. And that might be advantageous in clinical practice, especially in patients who we encounter who have large amounts of fluid. At 2 years, more than 60% of faricimab-treated patients achieved every 16-week dosing. And in fact, 80% achieved 12-week dosing and beyond, which would be a clear advantage in our clinical practice compared to the current agents we have in use. In terms of safety in neovascular AMD, luckily, faricimab was not associated with any higher rates of intraocular inflammation, and there were no systemic safety signals either. Of course, let's look to post-marketing safety of faricimab. Now it's been used more widely in the United States and in Canada, and we can see that as of July of 2022, more than 70,000 faricimab vials were distributed in the US. That number has increased dramatically now since 2023, and we have not seen any systemic or intraocular safety signals associated with faricimab. In summary, faricimab is the first bispecific that has been developed for retinal vascular diseases. It inhibits both VEGF-A and angiopoietin-2, and both of these agents have been implicated in angiogenesis and retinal vascular leakage. The pivotal phase 3 clinical trials of faricimab in both diabetic macular edema and neovascular AMD showed that faricimab was non-inferior to 2 milligrams of aflibercept. And more importantly, faricimab achieved these highly efficacious results with durability, allowing patients to go 12 weeks or every 16 weeks while achieving anatomic success and visual acuity gains. And finally, faricimab has shown an excellent safety profile, giving ophthalmologists and retina specialists confidence in using this drug for our patients. Thank you for allowing me to share this information with you.
Thank you, Diana. Let's see. Okay, so we're going to switch gears to actually share with you some real case, and I'm going to lead off with the presentations of the case that I have quite an interest and passion about pachyspectral disease, and Dr. Nicholas Fung is one of my Hong Kong U colleagues that helped me co-manage this patient. So this is a patient with chronic pachychoroid with PCV, polypoidal choroidal vasculopathy. And he was getting the regular aflibercept every 8 to 12 weeks for chronic fluid for many years. And he decided to move back to Hong Kong from Toronto, and he was continue on the aflibercept. But despite on every 6 weeks, the vision in the right eye is still relatively poor compared to the left. You can see on the OCT that his thickened choroid with this irregular peak PED with the double layer signed, which on the ICGA angiography confirmed the presence of branching vascular network with lay leakage on the fluorescein angiogram. And you can see the persistent subretinal fluid and the mild intraretinal fluid. And this continued to the same. And the problem is that what to do with this and whether you're going to extend it, so shorten the extensions to less frequent or switch to a different medications. So you can see the persistent fluid, and then at times the intraretinal fluid actually get significantly worse. So the decision was to try to something different, and the faricimab was tried. You can see with the faricimab, there is some reductions of the subretinal fluid. The intraretinal fluid probably hasn't changed that much from the treatment. So the treatment of polypoidal choroidal vasculopathy is quite difficult sometimes because anti-VEGF resistance is more common in this group of patients. And there have been some guidance you can get from the FRS2 and the PLANET study showing the either using the mono anti-VEGF therapy or combination therapy with PDT. And aflibercept has been shown to be more effective on the anatomical result for the treatments of particular PCV compared to that of ranibizumab. But what about the other types of anti-VEGFs, like such as faricimab? And in this study, 1 year result from the Japanese subgroup of the phase 3 TENAYA trial, where there's about 133 patients, 66 of them received faricimab, and 67, almost equal number, received aflibercept. And about a third of the patients have confirmed PCV on ICG angiography. And if you look at this anatomical result of the Japanese subgroup of the TENAYA compared to the pooled global TENAYA and Lucerne trial, you can see that the Q8 weeks of aflibercept and the Q16 weeks of faricimab are quite similar in terms of the efficacy in controlling the anatomical outcome. What is interesting is that at week 48, there's a higher proportions of patients in the Japanese subgroup. About 66% were able to extend to Q16 weeks compared to only about 45% in the poor global TENAYA and Lucerne trial. So why is the difference? One possible explanation is that there are higher proportions of patients of polypoidal choroidal vasculopathy in the Japanese subgroup. Like we have about 31% in that subgroup compared to only about 5% or 4.8% in the poor TENAYA and Lucerne trial. So maybe it is the PCV subgroup that drives the differences. However, of course, it is on a limited size, about 133 patients. And how about just the fact of there is pachychoroid, how that will influence the treatment outcome. And it is a short follow-up, 48 weeks. And there's also a very kind of regimented treatment dosing where you're comparing a fixed dose of aflibercept 8 weeks versus a faricimab treat and extend. What's good is that there's a Selwyn trial from South Asia looking at the efficacy and durability and safety of faricimab against in patients with PCV. So hopefully we will get some better idea whether this newer anti-VEGF may have a better outcome in treating patients with PCV. I'm going to ask Dr. Aboobaker to share his experiences in the management of using the faricimab. And just a brief introduction of Dr. Aboobaker. I think we stole him from South Africa, and he's one of the medical retinal specialists at the Toronto Retinal Institute. I think that he has been involved in multiple clinical trials, both as a principal investigator, as sub-investigator. But I just want to highlight that Dr. Aboobaker's passion for education and philanthropy has led him to be a co-founder of the Young Professionals Bursary, a scholarship aimed at assisting disadvantaged students in South Africa. So, Dr. Aboobaker.
Thank you, Wai-Ching, for that kind introduction. Good morning, everybody. I just want to share two of our cases of early sort of real-world Canadian experience with faricimab. The first case is an 85-year-old female who presented to us with wet macular degeneration in her right eye. You can see the subretinal fluid and some SREM. This was back in December of 2020. Visual acuity at that stage was okay, and we had started on aflibercept in that right eye. She had gotten 4 treatments with me and had done well up to the 6-week extension, failed the 8-week extension. And when I pulled her back to the 6 weeks, there was just this ongoing chronic subretinal fluid that, she was getting quite agitated about coming in to see me so frequently. So obviously in August, when we got approval for faricimab, I had the discussion. She was happy to switch. And she did well. She, within 2 doses, I was able to get her out to 8 weeks. Visual acuity had improved from the 20/40 to 20/30. And she's continued to do well on the drug following up. My last visit with her was out in March. You can see the anatomy looks much better than it was initially. Visual acuity is also on the up. And she was able to get out, well, I'll see her as soon as we get back from this meeting, but I've tried to get out to 10 weeks. So I'm hoping that she'll manage the 10-week extension. So she's happy with the additional durability that this drug is offering her at this stage. My second case perhaps shows a slightly different side to this. And this is a diabetic patient, a 58-year-old female who came in to see me initially about a year and a bit ago, presenting with DME that was worse on the left eye than the right eye. She was hesitant to jump into anti-VEGFs right away, so we discussed a session of a focal grid laser to try and give her some time to consider her options. My concern for her was that she had a visually significant cataract in the left eye that really had dropped her vision down to 2150. And I had a good chat with her about my concerns with the cataract surgery and the DME. You can see the large amounts of intraretinal fluid. And so eventually she came back. We elected to start treatment. And she had about 5 injections before her cataract surgery, which she did very well. She came back after the cataract surgery 2025. Still a good amount of retinal thickening. And you can sort of see there that intraretinal fluid was just ongoing. But thankfully her vision was good. She was quite happy, but quite motivated to sort of improve the retinal sort of imaging. So we switched in August again. And you can see there the path through her treatment. There's still ongoing IRF, but I feel the volume has sort of reduced. And so she's done quite well. She's quite happy. Actually missed a 4-week visit. She was out at 6 weeks back in April. And her anatomy sort of remained fairly stable. Not quite the sort of durability outcomes as the first case or some of the durability that Diana expertly shared with us from the phase 3 trials. But she's happy and doing fairly well. You can see on a color fundus images, the exudation has reduced since she's first come to see me through her treatment. So I just wanted to highlight these 2 papers from the same group out in Texas. They've used a similar methodology in both their DME and AMD papers. These were published fairly close together. And these were retrospective case control series looking at patients who had switched from aflibercept on a very tight sort of treat and extend protocol. They were aggressive treat to dry and then extend. Were patients who had better than 2200 vision, and these were more frequent flyers, as Sunir says. They only looked out to about 4 months in terms of the follow-up. The results here in the DME group show they had 51 patients, 24 on treatment with 27 controls. And the primary endpoint of this study was to assess the proportion of patients who had a CMT, a 4-month CMT of less than 300 microns. And you can see there about 37.5% of the faricimab group got to that endpoint. One in the aflibercept group got to that endpoint. In terms of visual acuity, greater than more than 2 lines, once again, faricimab around the 40% mark and aflibercept at the 11%. And very similar sort of findings in the DME cohort as well. This was about 28 patients in the treatment group, in the faricimab group, 27 in the aflibercept group, and you can see a similar sort of percentages there with some clinical significance. So I think the initial experience has been positive, and I think there's definitely a proportion of patients that are benefiting in the real world, and varying responses. But there's definitely a group that is doing well on the treatment. Thank you.
Okay, so thank you for all the presentations, and this is the time for questions period and the panel discussions. And please, there are a couple of mics in the audience, and you're welcome to use them. If you want to shout from your table, by all means do so. And
Preferably once you're standing up on top of it.
Shaheer, great cases. I wanted to ask you, when you're switching patients, let's say from aflibercept to faricimab, are you reloading them, or are you just automatically going to kind of a treat and extend personalized treatment interval?
Thanks, Diana. Yeah, that's such a great question. There's not a lot of data to sort of support exactly how to do the switch. I think in my patients, I'm sticking to the interval that they're on, and I'm not specifically doing a reload. Specifically, I'm sort of treating them till dry, and then extending from there.
I think that's what exactly I'm doing, because if a patient is already on, let's say, 6-week aflibercept or whatever agent, you don't necessarily want to go backwards and put them on every 4-week. And then I think most retina specialists are just using a treat and extend protocol if they're switching agents.
So a couple of things that I think is curious about us as a community of physicians. All the great data that Diana presented earlier was on treatment-naive people. But the cases that we're talking about are switchers. And you've seen that with our other drugs too. When we get a new drug, we tend to take these patients who are stubborn with their current agents and try the new stuff on them. So it kind of biases against the new drug. The other thing that happens, and I'm curious to hear you guys sort of just do a one-to-one switch and then pick up from there. Sometimes with the newer agents, I'll go back to monthly for a little bit because I just want to give this drug a chance to work. Because once I say, it didn't work for this patient, I close that door and tend not to reopen it. I don't always do that way, but many times I will.
So I think I also want to add to that, and I think Diana asked a very good question. I have to say that many of you know I've never been a big believer in the concept of loading, mainly because of the CAT data. Because the CAT data, they had one arm, obviously the PRN arm, where they didn't do a load, right? They went PRN right away, and but even in the monthly arm, after 1 year, they then allowed the patient to be randomized to then go to PRN after that. And they ended up with the same outcomes as the PRN from the beginning. So if 12 months of loading didn't make a difference, it didn't really seem to make a lot of sense to me that 3 months of loading would. But there's an important caveat when it comes to these newer agents. And one of the thoughts behind how, and assuming that you believe the Ang2 is relevant on how Ang2 works, that you might need to actually do loading to actually achieve that type of vascular stability. So I don't think we actually have the exact data because somebody needs to do a faricimab no load versus faricimab loading mechanism. But having said that, even though there's this theoretical reason to do so, I do what everyone else here, maybe Sunir, you do the monthly initially, but what Shaheer, I think, and Diana do, which is I don't load after the switch. I just try to continue on. But it's an important question that Diana raises, and we don't really have really strong data to support doing one or the other. Can I just make one other comment? Maybe it's going to lead into that. Just a big question that comes up a lot of times is also the timing of the switch. And obviously, in my talk, I sort of argued that there may be some clues that help us identify a patient who's likely to respond poorly. But I've been thinking about this a lot, and in terms of when we should switch patients. And a lot of times in the real world studies, and I think you highlighted some of them as well, Shaheer, in your presentation, but we've been participating in this study that Arshad Kanani has led, this Truckee trial of collecting real world cases. And we see that after switching, there's clearly additional drying. And I have cases where I switched them back and then you saw a recurrence of fluid. And so I have really good evidence that the switching actually made a difference. But the vision outcomes didn't show a big pickup in terms of vision, right? So you got the retina dry, you're able to extend them, but the vision didn't improve further. And that always made me think that the horse is already out of the barn. We should have switched earlier probably. And so I think there needs to be consideration for doing earlier switching And I wanted to see what others thought about that, because I've started to assess, like after my first injection, let's say I start with anti-VEGF, and there are a lot of times there's payer reasons that we would start with that I might even assess after the first injection, maybe even seeing back in a couple of weeks and see what kind of immediate response do you get. Because my experience has been patients who don't have that wow reduction after the first one. Yeah, you can get them drier with a few more injections, but those are the patients who end up not being able to extend so well. And then, maybe if you could switch them earlier on, maybe you'd actually could see an uptick in vision. But I'm curious to see what the rest of my distinguished panel thinks.
Diana, what do you do? Or do you load when you switched?
As I mentioned earlier, if I'm switching agents, I tend not to load because I don't want to shorten the interval that they've achieved. But I think in the United States and perhaps in Canada, we are restricted somewhat because we have step therapy, which is a fail first therapy that many of our payers require, where we have to choose usually the most cost-effective agent, which is off-label bevacizumab first, and then document that the patient had a suboptimal response before switching to another, let's say, on-label drug. And that might be disadvantage to the patient because we're not giving them the most optimal therapy that we would want to. And that could delay their visual improvement and anatomic gains.
I think in Canada, here in BC and Alberta, have the same kind of restrictions. And yes, I agree with that. Really limited the opportunity to get the patients on the right therapies. Sunir?
Yeah, so, I have the same constraints that Diana does in terms of choosing therapy. But as time has gone on, as Vas was suggesting, I think, sort of that persistence, let's keep going for 6-9 months and see how it goes, and then switch to drug 2. We end up leaving vision on the table. And I think getting the anatomy better earlier is likely to be advantageous. So I'm becoming more aware of that as time goes on.
So Shaheer, what do you do?
Yeah, I think Sunir and Vas make really important points, and I just want to emphasize those. When you're deciding how to start your treatment and who to switch, you're really looking at your frequent flyers, the sort of high need patients. And so, you may not get as much of that wow effect, but in the treatment naive group, I have seen a much better initial wow effect. So I think it's important in terms of patient selection as we sort of step into this and try to understand, where this drug has a role to play. In Ontario, we're very lucky, as you know, Wai-Ching, that we have access to the drugs, and we're not as restricted as the rest on the panel. Faricimab obviously is awaiting, a reimbursement on the public side. So we're looking forward to that, and I think we'll get a lot more data coming out from that.
Great.
I think it's going to be interesting as people do the health economic modeling, right? If you're doing fewer injections with a quote unquote more expensive agent, you might end up saving money. I will say that there's one payer in California that's made faricimab first line for DME, and that's their preferred agent because I think they've run their numbers and they found that they could save money using it. So it's going to be interesting. Again, I think, health economics is probably a good profession to go into because I think it's going to be important. But I think from our patient's perspective, I do think that as physicians, we need to advocate to be able to have a choice to push them to a different agent potentially earlier on, because I think that's where we're losing an opportunity to get the best outcome for the patient.
Any questions from the...
So, Vas, one of the things that I learned the other day is in the U.S., aflibercept costs around $2,000 U.S. Faricimab is $2,500 U.S. In Canada, faricimab is actually the cheapest at about $1,250 a pop. So I make the same argument to my peers, which is even though it's the most expensive drug, over time it'll be saving you money because I'll be able to extend you out. But here, the fact that it's so inexpensive compared to everything else, really I think makes it a pretty compelling argument.
While you have the mic, I want to ask you, like you share with us with quite a few emerging therapy that is on the horizon, and already some of them been approved. So of all those, which one do you think has the most promising therapy options that we should be looking at and consider?
Right. So that's a great question. And, it's part of the reason we do the studies is to tease that out. And part of the reason we have great research minds continuing to answer that is because we're still learning about the drugs. I think high dose of aflibercept would be a great option because that's going to be coming now. Everything else is still going to be a number of years out. But, my sense is that some of these other therapeutic targets are going to work. And it's now that we have Ang2, this is our first kind of time we've gone outside of the anti-VEGF paradigm, really in the past 2 decades. And so although that's still going to be likely the mainstay treatment, adding other therapeutic targets like tyrosine kinase inhibitors that have an anti-VEGF effect but potentially do other things as well, I think we'll start seeing more of.
Gene therapy, you mentioned, is it something that we'll be seeing in the AMD and DME?
That would be my expectation. There's multiple companies that are working in this space, and the data that we have right now, even though it's sort of phase 2 data, it's really compelling in reducing treatment burden. Part of the job will be to figure out which patients are going to be those home run patients, which ones aren't. Some of the reasons that you see variability may be due to antibodies that the body makes against the protein that's being made or against the adenovirus. So there's interest in looking at those neutralizing antibodies and kind of teasing that out. And I think just adding in that home monitoring, and as Vas had mentioned earlier, with the AI algorithms built into it, I think will really work well together as a pair. And that's sort of what we saw with anti-VEGF originally. We had OCT that was sort of out there and was kind of interesting. But then all of a sudden we had a therapy that worked together well with imaging technology. And I think you'll continue to see that play out just with the next generation.
Yeah, you mentioned AI. I want to ask Dr. Sadda. So how close are we using AI for diagnosing and helping management of DME and AMD? And what are the limitations?
So diagnosing, that might be a bit further out, but in terms of quantifying fluid, that probably not too far away, mainly because, we're expecting to see the results, for example, from a Tal's Home OCT study later this year. And assuming that those results are favorable and the device gets approved, it will come in with pre-built automated software, right? That'll quantify the fluid in the different compartments, giving us nanoliter results for the fluid. But mainly presenting to us the fact that the patient has had a recurrence. And again, we're going to have a lot of interesting choices to make because you can set your alert thresholds at different points and the like. It's going to be a very interesting space. So I think we're very close to having AI out there used in this kind of mode. It's kind of like, in a way, it's kind of screening for recurrent activity type of thing is probably what I suspect its approval is going to be. But in terms of more sophisticated things for diagnosis of neovascularization, things of that sort, I think the challenge in AI in general is like, who's going to pay for it? Like, I think a lot of these things can be done, but somebody has to bring that product, device, algorithm to the market. And then, if you're going to invest in doing that, because it requires a new prospective trial. So the regulatory agency, at least the US FDA, has said you can't improve an AI algorithm based on retrospective data. They have tremendous fear that some of that data might have somehow gotten into the model to train it in some ways. That creates a bias. So they've demanded that you have to have a new prospective data collection. That's expensive stuff, Wai-Ching. So, then you have to be able to make your money back. And that's what's still unclear in this kind of AI space, how to do that.
Thank you. So Amin wanted to ask questions.
Distant macular focal laser and PRP. So Shaheer, your case showed a type 3 CSME with a ring incident, very amenable to a simple laser and avoid multiple injections or reduce the injection load. And maybe Diana, you could comment on where PRP plays a role in neovascular DME.
Yeah, that's a very good question. Now with the anti-VEGF, are we abandoning the laser? Are we still using it in the appropriate patients?
Yeah, thanks, Amin. So as I presented, I still think there's a role, especially in a case like that, as you say, very amenable. And I had a discussion with the patient. I still use a grid laser or a focal laser when appropriate. I know the use has really reduced, and it makes sense because we've got really strong agents now that are, doing really well for our patients. But in those patients who are doing well visually, I know this patient had a significant cataract, so I didn't quite know what the vision would be after, and she did very well. And in that 2020, 2025, with some early DME that's especially non-central, I do tend to prefer to start with that and watch them closely as they usually worsen, unfortunately, and then get the anti-VEGF in. Diana, maybe you can touch on the PRP.
Yes. Well, in proliferative diabetic retinopathy, we do have some clinical trial data on using anti-VEGF monotherapy compared to pan-retinal photocoagulation in eyes with PDR, and that was the Diabetic Retinopathy Clinical Research Network Protocol S. And it's found that both can be very efficacious and safe for patients. Of course, with our current anti-VEGF agents, it does require frequent treatment if you're going to employ anti-VEGF therapy alone for the treatment of proliferative disease. We know that many patients, it's hard to come back regularly for treatment and follow-up. So that's why I think pan-retinal photocoagulation still has an important role in PDR, and it will help, reduce the VEGF load and keep our patients stable. I mean, all of us have patients who we've treated with PRP, and they've been stable for, 15, 20 years. So I think we still use it. Perhaps now I don't use as heavy PRP laser as I did in the past. So we're still preserving some of their peripheral vision. What about Vas and Sunir? Are you using PRP more?
See, I love PRP. I think that with these diabetic patients that they're just, as Diana said, Protocol S shows, yes, you can use either. But the reality is in the real world, a lot of these patients don't come back. There have been good compliance studies. I think you guys did one of those kind of compliance studies as well. At least I can say in Southern California, the compliance is not great. So PRP provides a great safety net. I would say that we're probably a bit more targeted in our PRP. I don't think we're doing as dense, sort of heavy kind of PRP. And I think you get the best of both worlds. You don't have as much compromise of the peripheral visual field, but you've got a safety net kind of in the background. So I think, we do plenty of PRP still. I will say our use of focal laser is plummeted, and I haven't done one in some time now. And part of it is because, there are two different things that are driving my approach and why I don't really do much focal laser, although I do agree in selected patients, it's still very reasonable. And one is that we obviously have studies like the RESTORE trial, which randomized patients right from the beginning to getting focal plus anti-VEGF versus, anti-VEGF alone. And there was no additional benefit that they observed in adding focal laser. Now, that was an all-comer population. So there were specific patients like the sursonate patient off to the side that you showed. Probably that's the case. I don't think that's been dissected well enough. But at least in that study, when they compared focal and diffuse, they didn't really see a big benefit. So that's one thing. The other thing is obviously in protocol. I'm going to get it. Protocol V. The fact that you can observe these patients for some time, even with center-involved DME, as long as once they have vision loss, you start to treat them, and you still end up with the same vision outcomes. That to me is kind of reassuring. That's why I stopped treating these non-foveal DME patients, because if foveal DME with good vision you can observe, then why am I treating non-foveal? And the good news is, unlike PDR, where maybe the patient is having significant disease, they're not noticing it. Patients usually will notice this and they will come in. So the DME patient who's non-compliant is more likely to come in at least when they have vision loss compared to a patient with PDR who's non-compliant. So that's sort of the sum of the different components that's gone into my logic for why I still do lots of PRP, but not much focal laser.
Yeah, I'm pretty much the same. Nothing really different to add to that.
Any questions from the audience? John, you've been quiet. Yes. You're not a quiet person, I know.
I have a question for Vas. You talked in your own question about you talked about biomarkers, easy. That's you're really talking about visual acuity outcomes. Whether they're poor or top. Do you have biomarkers? I know you talked about focal reflectors on that. Are those, strictly speaking, used anti-VEGF? Are these anatomical markers for response?
Yeah, so the question was about just which of the markers, just for those who might not have heard on the other side of the room, the question was about which markers may be relevant for vision outcomes and which markers may be relevant more for treatment response in terms of anatomic reduction to anti-VEGF monotherapy in particular. And yeah, I know I went through a bunch of them pretty fast. And so, yeah, so drill and EZ are primarily markers of visual outcomes, as you suggested, John. That is what they show. That they really don't have so much to do with anti-VEGF monotherapy versus other mechanisms. Although there's some data to suggest that perhaps targeting other mechanisms or at least getting the retina dry faster can lead to some reversibility of these. So I think that's an interesting observation. But yeah, the hyperreflective dots and the subretinal fluid, those are the two that I highlighted, seem to be biomarkers associated with poorer response to anti-VEGF monotherapy. It doesn't mean that they can't respond, but they seem to be generally associated with a poorer response. And so poorer anatomic response usually ultimately will lead to poorer vision outcomes. So probably it's both for those particular biomarkers. But I point them out mainly in the context of this discussion in terms of patients that you might be thinking that, I don't want to wait too long to switch them, or if you have the ability, like depending on whatever the rules are in a particular province, maybe those are the patients that you consider starting on a different agent. Because I do think that we need to have a different switching regime. I really feel like these step therapy things are harming patients. And I don't think that, that's actually what the DRCRnet really needs to help with, is to arm us with good data to show that step therapy is bad. Because I think it could be bad. And I'd like to know, if it's not bad, then that's great, then we will learn something. But the fact that we haven't proven that very well yet, in my opinion, is concerning.
So, Vas, if I can, following up on the step therapy, DRCRnet, of course, just came out with a protocol, I think AC, that said that, well, you could start with bevacizumab, and then over time, 70% of people would have switched to aflibercept, which is the alternative drug that they used. And there's this debate going on in the U.S. about sort of the cost utility of that. The DRCRnet, my impression reading the paper was that they were sort of like, hey, there's nothing bad with doing it this way. And some patients do great with bevacizumab, and it'll save everybody money. And for those who eventually got switched over, well, that's what they needed, and that's fine too. But then other people are saying, well, no, but if you amortize those costs over time, all of a sudden that number changes. What's your take on that?
Yeah, thanks for pointing out AC. That's probably the best protocol that's actually evaluated this question. And unfortunately, it's, by the time these studies are done, then you have the next generation of agents. So you don't know. So, and the like. So that's one limitation of protocol AC. But yeah, my thought about that is that the other challenge is probably it is true for all comers. You step therapy may not be unreasonable, and it's hard to find the potential harm of step therapy in an all-comer population, because there are plenty of patients who are going to respond well to bevacizumab. In fact, it's just those patients who have maybe these biomarkers associated with a poor response or something of that sort that I think are harmed, and that can be lost in the noise. Also, of course, we have a little bit of a better margin with DME compared to neovascular AMD. So that's another thing, because obviously, we can tolerate edema for a while from protocol V before the vision loss becomes permanent. So that's the tricky aspect. I do think, though, that I think that these health economics analyses are pretty important. I think that I think if you can isolate that group of patients, to decide early on that this is not going to be a great responder and treat them early, that's where I think you'll have cost savings as well as better outcomes for patients. And I think that requires some very carefully designed trials.
Thank you. I want to ask the panel about subretinal fluid. Like how much subretinal fluid you tolerate, and I think Dr. Sadda mentioned that it is actually not a protective, as it's been kind of discussed. There's quite a bit of controversy on that topic. So maybe we just start down the road. I start with Diane.
I agree with Vas. I try to eliminate all types of fluid, including subretinal fluid.
Yeah, I kind of already gave my bias that I don't think that subretinal fluid is a positive thing. I think the reason that there's subretinal fluid may be a positive thing if it's a type 1 membrane. And so the studies that have shown that it's positive, quote unquote positive, more or less, there are patients who are receiving treatment. So the idea is you're treating it and you're not letting it get out of hand. But the fact that it's there may be indicator of something else that's protective, which is the type 1 membrane, is my philosophy. So yeah, I don't tolerate fluid.
Yeah, I'm exactly the same. I also believe it's still a sign of activity. I'm aggressive treated dry. And in patients who have been with me for a couple of years, I sort of bring them in at the 2-week mark at some point just to show them, well, here's the benefit that the drug is actually showing with reduced fluid, and why they still at the 4 weeks, because between the 2 to 4 weeks, that fluid is sort of building up over time.
And I'll same but a little different. I'll try to get rid of fluid whenever I can, and I'm fairly aggressive about trying to get rid of it. But there's some patients who have this tiny little bit of SRF, and they're have healthy RP, they're doing pretty well. They're at 7 weeks, let's say, with a tiny bit of SRF. To get rid of it, I have to bring them back at 4 weeks. So then the practical considerations come in of what's the value proposition for that patient. And so there's certain exceptions to the rule. So I'm not an absolutist about it.
Yeah, I think actually Sunir made a good point that I didn't highlight in my presentation because of the time. But my colleague David Saraf in particular has been talking about this quite a bit, and we've published some papers together on the concept of non-neovascular fluid. So not all fluid. I kind of hinted at it about intraretinal fluid. I said some of that may be degenerative, but the same actually applies to subretinal fluid. Subretinal fluid can arise from other mechanisms. So Wai-Ching showed a beautiful pachychoroid spectrum disease, and one of the challenges in pachychoroid spectrum disease is sometimes there can be multiple mechanisms, right? You can have fluid accumulating because of the neovascularization and exudation, but it could also be because of pump failure. So RP pump failure is an important contributor, just like Mueller cell injury is an important contributor to degenerative intraretinal fluid. This is an important contributor to this type of non-exudative subretinal fluid. So I tried to get rid of it. And one of the tests that I like to do is you treat the patient, you bring them back a week later and see what they look like. And if they still have a fluid, or they haven't had a substantial reduction in fluid, I'm assuming that fluid is there due to other mechanisms. And so in that situation, I may tolerate it, or I may try to target those other mechanisms. And for example, in a pachychoroid spectrum patient considering PDT.
So, any questions from the audience? We're down to the last few minutes. So I think it's been wonderful symposium, wonderful panelists. As a moderator, I don't need to do very much. I think they might get taken away right away. I think a very engaging expert. I think we have heard about the new kind of emerging therapy, some of the new long-lasting anti-VEGF gene therapy and sustained delivery devices. The role of dual actions, Ang2 pathway, how it impact patients, the outcome in DME and neovascular AMD. With the OCT biomarkers and the different imaging biomarkers, we probably in a better positions to predict treatment response and maybe even personalize the patient therapy. I think that's probably the direction that we're going to. And so I just want to thank the panelists and for sharing their expert opinion, and thank you for coming in this early in the morning to participate in this symposium. I think there is a slide here for the evaluation. So I think you'll get that by email for the certificate of attendance and your evaluations. And please do put in your evaluations because it will help us to improve our programs. And otherwise, enjoy the rest of the day in the COS, and hope to see you next year in Toronto. Thank you.